Hello everyone, welcome to lecture 4 on biological classification. So in the last lecture, we learned about the kingdom Monera. So we learned that in kingdom Monera, all the prokaryotes were included in that kingdom. That includes bacteria, archaebacteria and mycoplasma. So we learned about the characteristic of that kingdom in the last lecture. And in today's lecture, we will proceed with another kingdom, that is the kingdom Protista. So we know that in the biological classification, the classification system that was given by R.H. Whittaker, so all the organisms were divided based on various characteristics. So the first kingdom that was defined was the kingdom Monera, that includes the bacteria, archaebacteria or the other prokaryotic cells, while the other eukaryotic cells were kept into the separate kingdoms, out of which the first kingdom is the kingdom Protista, and the other remaining three kingdoms were kingdom Plantae, Fungi and Animalia. So kingdom Protista is the first kingdom of eukaryotic cells that were defined after kingdom Monera. It includes algae and protozoans, and we will learn in details about kingdom Protista today. So while we learned about the 5 kingdom classification that was given by R.H. Whittaker, so one of the limitations was that the kingdom Monera, so one of the limitations was that the kingdom Protista had a problem with the fact that it includes many eukaryotic cells. And sorry for the disturbance. So when we were studying about the 5 kingdom classification by R.H. Whittaker, so we learned that one of the problem with the kingdom Protista was that it included organisms that were not of very similar types. So basically, however, since the NCERT defined all the kingdoms on the basis of the 5 kingdom classification only, so therefore we will be learning the kingdom Protista according to that course. So the Protista, as I mentioned, it is a catch-all group for any eukaryote that does not fit either into the kingdom Plantae, that does not fit either into the kingdom Fungi, and that does not fit into the Animalia. So basically, the eukaryotic cells that cannot be considered as plants, animals, or fungi were put into the kingdom Protista majorly. So basically, the characteristics of the organisms of kingdom Protista are very mixed type. It is a very heterogeneous group. So we will learn about the characters of kingdom Protista. First and the foremost very important character is that the organisms of kingdom Protista are eukaryotic in nature. So we learned about prokaryotic cells and eukaryotic cells in the last lecture. So prokaryotic cells are the cells that are not covered with that does not contain cell organelles that have the membrane. So they lack membrane bound cell organelles, while the eukaryotic cells are the cells, or the eukaryotic organisms are the organisms that contain the cells having a membrane bound organelle and a true nucleus. So basically they have membrane bound organelles and a true nucleus. Similarly, as I mentioned, that since it is a very heterogeneous group, therefore the organisms can be both unicellular as well as multicellular, and they may also exist in the form of colonies. So basically, they also exist in the form of colonies. So the organisms here can be unicellular, multicellular, or can exist in the colonial form. Also, when I talk about the nature of their nutrition, so they may be heterotrophic, that is, they may be dependent upon other organisms for their food. They may be autotrophic, that is, they may be photosynthetic and can survive, can make their own food using sunlight or by various other methods. Like they have their own chlorophyll, using which they can synthesize their own food. Or they may be mixotrophic. So mixotrophic means they can be both autotrophic as well as heterotrophic depending on their condition. So protists exhibit various modes of nutrition. Some are photosynthetic, that is, they are autotrophic, while some are heterotrophic, they obtain nutrients from other organisms, and some are mixotrophic, that is, they are capable of both autotrophy and heterotrophy. The next is the reproduction. So protists reproduce through various methods. we see that in case of bacteria, mostly the most common method was the asexual reproduction. And uh, while in Protista, they reproduce through various methods, including binary fission, which is also a kind of asexual reproduction. Instead of binary fission, they may undergo multiple fission, they may undergo conjugation, or they may undergo spore formation and sexual reproduction in some cases. And this specific method depends on the type of protist of which we are talking about. So motility, that is whether they are able to move or not. So many protists can move, and the movement is facilitated by the structures like flagella that allows or that helps in the movement of the protista, or it may be facilitated by the presence of cilia or pseudopodia. So pseudopodia are false feet, we can say, or false finger-like projections that we will see later in the lectures is present in some class of kingdom protista. And these pseudopodia not only, basically these pseudopodia helps in engulfing the food. So this is these are some of the characteristics. Beside this, the habitat. As we know that protists are very heterogeneous in nature, therefore their habitat is also varied. So they can be found in various habitats that include fresh water, marine water, soil, or they may be present as symbionts in the other body organisms. So basically, they may be present as symbionts in the bodies of the other organisms. For example, we know Plasmodium. So Plasmodium acts as a parasite, we can say, and is present or can infect the RBCs. So malaria, which is caused by Plasmodium, is a kind of protist. So Plasmodium that causes malaria is a protist. So it can habitat inside the body of the other organism. The next is the cell wall composition. So cell wall composition varies among different groups of protists. For example, we will learn later about this also. So for example, some may have cellulose-based cell walls, while others may have silica-based or calcium carbonate-based cell walls. So the cell wall composition also varied. It can be cellulose-based, it can be silica-based, or it can be calcium carbonate. Diversity, we know that kingdom protista is highly diverse. It encompasses any organism which does not fit into kingdom Plantae, fungi, or animalia. Therefore, it includes algae, protozoans, and slime mounds. And they exhibit a varied form of structures. They exhibit variety of forms and structures as well. If I talk about the ecological roles, so just like we talked about the ecological roles of Monera also, where they helps in decomposition, they helps in nitrogen fixation for the plant. Similarly, protists also have their own ecological roles. So protists play crucial ecological roles, contributing to processes such as nutrient cycling. So many of them are aquatic, and therefore they help in the nutrient cycling in the aquatic ecosystem, as well as they act as the primary producers in the aquatic ecosystem. So we know that many of the protists are autotrophic as well, that is, they can synthesize their own food. Therefore, they act as the primary producers in many ecosystems. So these are some of the characteristics about the kingdom protista. And while learning about this characteristic or while going through it, we get to know that actually the kingdom protista is diverse, highly diverse, be it on the basis of the mode of nutrition, mode of reproduction, the flagella or the basically the structures that facilitate the mobilities. All of these are very diverse in different classes of kingdom protista. So although their kingdom protista can be divided into many divisions and many classes, but we will talk about only limited ones that are described in class 11th NCERT. So firstly, we will talk about the division Chrysophyta. This division includes the organisms, that is diatoms and golden algae, which are known as desmids. And as I mentioned, they are found in fresh water as well as in marine environments. These are microscopic eukaryotic cells, and these organisms, the organisms that belong to kingdom protista division Chrysophyta, does not have flagella. Since they do not have any flagella, Therefore, they are non-motile because they cannot move. Because flagella helps in movement of the organism. So basically, they do not have any motility appendages that helps in the movement of the organism. And therefore, due to the absence of flagella or any such motility appendages, these organisms are non-motile and are usually they float passively in water currents, and therefore they are known as planktons. So basically, planktons are the organisms that float passively in water currents, right? So they appear as planktons. Now they form a key group of photosynthetic protists. So they are photosynthetic in nature. The organisms that belong to division Chrysophyta are photosynthetic. They are unicellular algae that have unique glass-like wall made up of silicon dioxide, which is embedded in an organic matrix. So it is very important to understand the structure of the cell wall, or we can say the composition of the cell wall, because it is really important when we talk about this cell wall composition in the kingdom Protista. So the cell wall of the organisms of kingdom Chrysophyta, of the kingdom Protista division Chrysophyta, is made up of silicon dioxide. So it is a silicon dioxide cell wall that is made up of, and due to which it is really hard in nature and it is difficult to destroy. Therefore, it helps in the protection of the organism from the predators as well. They stored food in the form of leucosin and fats. So basically, there are some points that we need to remember when we talk about different kingdoms. Firstly is their cell wall composition that may vary in different divisions of the kingdom Protista. Also, the type of the way they stored food. So how they stored food in which form is important to remember, and examples we need to remember for different divisions, right? So they stored food in the form of leucosin and fats, and in diatoms, diploid nucleus is present, right? So 2N, it's diploid nucleus which is present, and diatoms, as I mentioned, that they are photosynthetic in nature. They are key producers in the aquatic ecosystem. They have pigments like chlorophyll A, chlorophyll C, and xanthophyll, right? So, which is also known as fucoxanthin. And due to these pigments, they appear golden color. So, due to the xanthophylls or fucoxanthin which is present, the color of these organisms is usually the golden in nature. So, a very important thing is to understand the structure of diatom, right? So, the cell wall, as I mentioned, cell wall, which is also known as shell of diatoms, is made up of cellulose in which silica particles are embedded at many places. So, basically, it is made up of silicon dioxide, and the structure of diatom cell wall is really very interesting because it is just like a shoe box, we can say, or a soap box lid, right? So we know when we see a shoe box, we see a box which is covered with a lid. Similarly, this is what the structure of a diatom looks like. So this silicated cell wall is called a shell or a frustule. So this is the structure of diatom. So basically, the silicated cell, so this is the structure of the diatom. So this silicated cell wall is called as the shell or frustule. So this is known as the frustule. Now, as I mentioned, like we have a shoe box. So one is the box, the other one is the lid. So if I consider this as a shoe box or a soap box with a lid, we can consider two parts, right? One is a box and the other is the lid. So the lid part is known as the epitheca, that is the upper part, and the lower part is known as the hypotheca. So it's epitheca and hypotheca. So their cell wall, as I mentioned, is made up of two halves which are arranged like the lids of a soap box or a shoe box. Their cell wall have silica in very large quantities, as I mentioned, and due to this reason, their cell wall is really hard. And even if they die, even if the organism die, it is difficult that to destroy that cell wall, right? Therefore, their cell wall does not get destroyed even after the death and keeps on accumulating at the bottom of the sea. And such huge rocks are formed due to the accumulation of these cell walls, silica cell walls at the bottom of the sea, which are known as the diatomaceous earth, right? So a name has been given to this, which is either diatomite, which is the huge rocks which are formed due to the suspension, or we can say not the suspension, but due to the deposition of the silica cell walls of the diatoms at the bottom of the sea. So diatoms, diatomaceous earth, or Kishilgo, right? So these are these are the terms that we have given to the huge rocks which are formed at the bottom of the sea. So this was about the structure of diatoms. So it is the structure can be more clearly seen from this figure. So we can see that we have an upper part and a lower part. So we have an upper part and a lower part, right? The lower part is fixed in just like a box, and the upper part acts as a lid, and the upper part is known as the epitheca and the hypotheca. Lower part is known as the hypotheca. So basically, diatoms reproduce mainly through asexual reproduction, which is the vegetative form of reproduction, and it also undergoes sexual reproduction. So when a cell divides, an interesting form of division takes place. So when a cells divide by the process of mitosis, so for epitheca, a newer hypotheca will be developed, and for hypotheca, a newer epitheca will be developed, right? And every time, whenever the newer part which is formed is always the smaller as compared to the older part. So we can see that as it keeps on, so it will produce firstly a daughter cell of the same size, and the other daughter cell which will be produced will be of smaller size, right? The one in which the hypotheca remains the same will be of smaller size, while the one in which the epitheca remains the same will be of the same size as the parent cell. So if this keeps on happening, there will be a time when the size of diatom will keep on decreasing, because we know that with every division, the size of the daughter cell is decreasing, right? We are getting one daughter cell which is of smaller size. Now when this will divide, we will further get a daughter cell that will be further smaller than this, right? So therefore, there will be a stage after which it cannot undergo this vegetative reproduction. So statistically, the size will keep on decreasing, and when the smallest diatom reaches a minimal size. So when a minimal size is reached, then at that stage, instead of asexual reproduction, sexual reproduction is required to gain a cell of maximal size. So in that case, sexual reproduction takes place in case of diatoms. However, in rare cases, there is a vegetative cell enlargement also, which is rare. But in usual cases, when a diatom reaches the smallest minimal size, it has to undergo sexual reproduction to regain its larger size. So, we know that basically it undergoes asexual reproduction, but at some point of the life, it has to undergo asexual reproduction when it reaches the smallest minimal size. The next class is the dinoflagellates. So, similar to diatoms, they have a protective cellulose plate which are bound by a single cell of dinoflagellate impregnated with the silicates. So similar to diatoms that has the cellulose plates and that in which the silicon dioxide is present, dinoflagellates also have the protective cellulose plates, and these cellulose plates are impregnated with the silica particles. So they are impregnated with the silicates. Typically, as the name also suggests, they have two flagella. So instead of one, they have two flagella. Diatoms were mostly amotile, while the dinoflagellates are motile due to the presence of flagella, right? So typically, they have two flagella. We can see that one lies in the longitudinal groove with its distal end free. We can see this. The other one is not visible in this diagram, while the other lies in a transverse groove that encircles the organism. So yeah, it is visible here. So we can see here. So one is the longitudinal one, the other one is the transverse one. So you can see it here, right? So these are the two flagella which are present. So the one is the longitudinal that act as a radar, and the other encircles the organism, which is a transverse flagella, and the beating of transverse flagella causes the cell to spin as it moves forward. So it's move forward with the spinning movement. So it helps the cell to spin as it moves forward. So that is the function of transverse flagella. So the transverse flagella and the longitudinal flagellum helps in the movement. So longitudinal flagellum acts as a rudder, while the beating of transverse flagellum helps the cell to move forward, causing it to spin. The chloroplast of dinoflagellates vary in color from yellow-green to brown because in addition to chlorophyll A and C, they contain carotenoids. So just like the diatoms have xanthophylls, these dinoflagellates have carotenoids, and based on the color of the carotenoids that are present in these dinoflagellates along with these chlorophyll A and C, they are of different colors. So the chloroplast of a dinoflagellate vary in color due to the presence of various carotenoids, which are colored pigments. So it is really important to, so what is the point to remember here is that dinoflagellates are the organisms with two flagella. One is longitudinal, one is transverse. Function of those flagella and what are the pigments that are present. So pigments that are present is chlorophyll A, chlorophyll C and carotenoids. And how dinoflagellates are similar to diatoms is that they have silica in their membrane. They have cellulose plates and basically they have chlorophyll A and chlorophyll C just like the diatoms. But in diatoms we have xanthophylls. Here we have carotenoids. So dinoflagellates mainly reproduce asexually by the process of binary fission. There is no soap box or lid hypotheca-like structure. They simply reproduce through binary fission asexually. Sexual reproduction is rare, while in case of diatoms, we see that at some stage they have to undergo sexual reproduction when they reaches a minimal size, right? They stored food in the form of starch, while the diatoms stored food in the form of leucoferrin and oils. The other thing to remember in dinoflagellates is that some of these species are well described in NCERT, where their properties have been described. So, for example, some of the species of dinoflagellates like Noctiluca and Goniolix can have bioluminescence. So, Noctiluca and Goniolix have the property of bioluminescence. So, by this we means that they have the property of producing light on their own. So, there are certain pigments that are present inside these organisms. So these pigments, when came into the contact of some light, they produce these pigments under certain conditions produce light. So basically, the organisms have the capacity to produce the light. Therefore, the process is known as the bioluminescence. So these organisms, that is the Noctiluca and Goniolix, can have bioluminescence in them. That is, they have the tendency to produce the light. When dinoflagellates such as Gymnodinium brevis and Goniolix can increase in number, so they are red in color, therefore they causes a phenomena which is called as red tide, right? So when their number increase, the tide appears to be red in color, and therefore the phenomena is known as the red tide. These dinoflagellates, Gymnodinium and Goniolix, produce powerful neurotoxins. These powerful neurotoxins are very dangerous and they can lead to massive fish killing. So when they release these toxins into the environment, the water becomes so toxic that it leads to massive fish killing. And also these toxins enter into the food cycle and can harm humans as well if the infected fish is being eaten by the humans. So these toxins are that dangerous. So we talked about certain examples in dinoflagellates like Noctiluca, Goniolix and Gymnodinium. Noctiluca and Goniolix have the property of bioluminescence. That is, they have the tendency to produce light. They can produce light. Similarly, Gymnodinium and Goniolix are the dinoflagellates that causes the phenomena of red tide. And they produce toxins, neuronal toxins that harms the neurons and basically lead to the massive fish killing released into the environment. They are really harmful for the fish and also for humans, and they can enter into the food cycle through the fish. The next class is the euglenoids. So till now we learn about Rhizophyta and dinoflagellates. Dinoflagellates belong to division Pyrophyta. Now we have euglenoids. So euglenoids are very interesting, as in some aspects they are similar to plants, while in other they are similar to animals. So in earlier times, zoologists have put euglenoids into animal kingdom, while botanists have considered them as plants. And so there was a confusion whether the euglenoids are plants or animals, and they are now currently in the kingdom Protista, as they have the characteristics matching with both plants and animals, but not the characteristics unique either to the plants or to the animal. Therefore, they are placed in the kingdom Protista. So basically, in the earlier times, euglenoids were placed in the plant kingdom due to their photosynthetic ability. So the euglenoids have photosynthetic ability, due to which they were placed in the kingdom plants, in the kingdom Plantae. However, due to the absence of cell walls and animal-like nutrition, some scientists place them in the animal kingdom. So that is what I was mentioning, is that euglenoids are interesting in the fact that they resemble both plants as well as animals in certain cases. So they have the photosynthetic ability of the plants, while they do lack cell walls, due to which they resemble animals, and people have tried to put it in the animal kingdom as well. But now, according to the 5 kingdom classification that was proposed by R.H. Whittaker, they were placed in the kingdom Protista. They are free-living organisms in freshwater lakes, ponds, etc. But sometimes they are also found in damp soil and brackish water. So as I mentioned, Protista are found in various habitats, in varied habitats. So euglenoids are present not only in fresh water or ponds, but sometimes they are also found in the damp soil and brackish water. These organisms are photosynthetic in the presence of sunlight. So in the presence of sunlight, they show characteristics. Due to their photosynthetic ability, they are able to produce their own food. However, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms, for example, euglena. So euglena is an example of euglenoid group. So euglena has the tendency to show mixotrophic nutrition, that is both autotrophic and heterotrophic. So in the presence of sunlight, they undergo autotrophic mode of nutrition, while when they are deprived of sunlight, they undergo heterotrophic mode of nutrition, where they predate on other smaller animals for their food. So basically, euglenoids can undergo both the can switch between both the modes of nutrition, that is between the autotrophic and heterotrophic. So as I mentioned, that the stored food in the case of dinoflagellates is different in the form of starch. In the case of diatoms, it is mainly in the form. So in the case of diatoms, it is mainly glucosin and fats. While if I talk about the euglenoids, it is mainly paramylum and fat that act as a stored food for them. So in case of euglenoids, it is paramylum and fat that act as a stored food for them. Reproduction takes place asexually by longitudinal binary fission. So there are 2 binary fission that may happen. One is along the transverse axis, that is the organism is dividing transversely into the 2 halves, the first half and the second half, right? While there is the another division that may happen, the one is the longitudinal, where the organism is divided longitudinally into 2 halves, the first half and the second half. So basically, in this way, 2 organisms are produced, longitudinal binary fission or the transverse binary fission. So in euglenoids, it is the longitudinal binary fission that happens, and the organisms reproduce asexually through longitudinal binary fission. So the organism divided longitudinally into the 2 different organisms into 2 daughter cells. Example includes euglena, as I mentioned. So it is a very common example of the deficient euglenoids. The structure of euglenoids can be understood with the help of the example of euglena, as we can see in the right hand side diagram. So they are surrounded by a cell membrane. They do not have a cell wall. So as I mentioned, they lack the cell wall just like animals, and they are surrounded by a cell membrane. So they are surrounded by a cell membrane which is made up of lipoproteins, and this cell membrane is covered with a pellicle, thin layer which is known as the pellicle. So the cell membrane is covered with a pellicle, and and they lack the cell wall. They have a long flagella, and at the anterior end, cavity is present which is known as the reservoir. So this is the anterior end, this is the posterior end. So at the anterior end, a cavity is present which is known as the reservoir, and flagellum is originated from the base of this reservoir basically. So the origin of this flagella we can see from here. So this is from the base of the reservoir. So the cavity is known as the reservoir, and the flagellum originates from the base of this reservoir. Euglenoids have only one functional flagellum and one non-functional flagellum. So we can see that in euglenoids there is a short flagellum which is present and a long flagellum which is present. So the one is functional while the other is non-functional. So it has two flagellum, the one functional flagellum and the one non-functional flagellum. Also, one eye spot is present in the anterior position. It has an eye-like structure basically. When we visualize it, it seems that it's an eye to it, since it is known as an eye spot. So one eye spot is present at the anterior position, and they have a contractile vacuole. So the function of the contractile vacuole is to maintain the osmolarity. That is, it helps in the osmoregulation. So we have two flagella. One is the short flagellum, one is long flagellum, one is functional, one is non-functional. We have eye spot-like structure that contains a photoreceptor that helps in the detection of the light, right? Then we have contractile vacuole. These contractile vacuoles help in the osmoregulation. That is, it helps in maintaining the osmolarity inside the cell. So we know about the osmolarity, right? So it depends on whether it is present on the hypotonic solution, whether it is present in the hypertonic solution. So when an organism is present in a hypertonic solution, the water will move out of the cells and the cell might shrink. While when it is present in the hypotonic solution, the water will rush into the cell of the organism, and if we do not remove that organism, it will burst due to the large amount of intake of the water, right? So it helps, this contractile vacuole helps in maintaining that osmolarity. It helps in the osmoregulation. And euglenoids have a haploid nucleus. So we mentioned that earlier the diatoms have a diploid nucleus, that is 2n type. The nucleus in this case is an haploid type, and since it is a eukaryotic cell, the nucleus is covered with a nuclear membrane, and it is not a naked nuclei just like bacteria. So it is covered with a nuclear membrane. It consists of a nucleolus also, and they have the chloroplast that is present in these organisms that helps in synthesizing the food in the presence of sunlight, and the pigments, these pigments which are present in the euglenoids is similar to the ones which are present in higher plants. So that was the reason they were earlier kept in the kingdom Plantae. Due to these reasons, there were some reasons due to which they were kept in. People have tried to kept it in the kingdom Animalia, but now we know that they are neither animals nor plants, but they are a separate group and are now kept in the kingdom Protista. So chloroplast, the pigments that are present is chlorophyll A, which is similar to dinoflagellates and diatoms. Chlorophyll B, which is similar to higher plants. In diatoms and dinoflagellates, it was chlorophyll C that was present, while in euglenoids, it is chlorophyll B, and the xanthophyll is present that help. So basically, these pigments help in the photosynthesis in the presence of sunlight, and these pigments are identical to the higher plants. So due to these similarities with the higher plants, as I mentioned, they were kept in the kingdom Plantae also. But If asked in the examination, euglenoids belong to kingdom Protista. What are the basic characteristics? They lack a cell wall, they have a cell membrane which is made up of lipoproteins. They have a structure which is known as reservoir, from the base of which there are two flagellum that originates. One is the long and one is the short flagellum. They have contractile vacuoles. These contractile vacuoles help in the osmoregulation, that is maintaining the osmolarity of the cell. They have a nucleus which is surrounded by a nuclear membrane, which is a eukaryotic cell. They have chloroplast. Chloroplast pigments are chloroplast A and chlorophyll A and chlorophyll B, and xanthophylls, which are similar to higher plants. The next group is the protozoans. So now we studied about dinoflagellates, diatoms, euglenoids. Now we will learn about protozoans. So protozoans are also broad class under the kingdom Protista, and the term protozoan can be somewhat ambiguous as based on different people have defined it differently. So some restrict this term to the unicellular heterotrophic organisms that ingest their food, while this can also mean the unicellular or colonial eukaryotes. So in that case, if we go by the first definition, they are heterotrophic in nature, that ingest their food, that is, they acquire their food and then they ingest their food. While if we go by the other definition, where it means any unicellular or colonial eukaryote, so in that case, protozoans may also include the photosynthetic, heterotrophic or mixotrophic organisms. So different people have defined it differently, and therefore the term protozoan is still ambiguous. Most protozoans have some form of locomotion, so they have locomotory appendages, well-developed locomotory appendages, like they have flagella, they have pseudopodia, they have cilia, and those that don't locomote tend to be parasitic. So basically, they can be both motile as well as immotile. So on the basis of their motility, for the motility or for the locomotion, they can have flagella, pseudopodia or cilia, while if they are or they do not have the tendency to locomote, they tend to be parasitic. So based on that, at one time, zoologists classified protozoans as animals, considering them to be heterotrophic in nature. But if we go by the other definition, where the protozoans means unicellular eukaryotes, that means that it can include the photosynthetic organisms as well. So therefore, it is difficult to classify them either in the kingdom Plantae and Animalia, and therefore they are kept in the kingdom Protista. So even from the definitions of the different divisions that are present in the that are kept in the kingdom Protista, we can consider that the heterogeneity of the kingdom Protista is diverse, and people have earlier tried many times to place these organisms into try to keep these organisms either to the kingdom Plantae and kingdom Animalia, but due to certain differences which are not exclusive to the particular kingdom, like kingdom Plantae or to the kingdom Animalia, they have to put them into the other kingdom, which is the kingdom Protista. So just like euglenoids, people have tried to put protozoans as well into the kingdom Animalia, but it is not exactly similar to kingdom animals in many aspects, and therefore it has now been put into the kingdom Protista. So we will learn about different protozoans. The first is the amoeboid protozoans. So amoeboid protozoans are the protists that move and also ingest their food with the pseudopods. We know that different protozoans have different modes of locomotion, appendages for the locomotion, that is either flagella, pseudopods or cilia. So the amoeboid protozoans have pseudopods. So we can see pseudopod here. Pseudopod. Pseudo means false. So the word pseudo here means false, right? Whenever the word pseudo has been used, it means false. And the word pod has always been related to the appendages. So these are false appendages or false finger-like projections that helps in the locomotion and in the capturing of the food. Hundreds of species of amoeboids have been classified. And when amoeboids feed, the pseudopods surround and phagocytize their prey. So basically, amoeba feeds through the process of phagocytosis. That if it finds a food here, suppose it finds a food here, suppose it's a food particle which it finds here. So this pseudopodia will come and extend and will then trap the food and will take it into the cell, keep it inside the cell. So it will undergo phagocytosis. So the amoeboids, so when amoeboid feeds, the pseudopods surrounds and phagocytize their prey, which may be algae, bacteria, or other rapist. Digestion then occurs within the food vacuole. So food vacuole contains enzymes that helps in the digestion of the food. Freshwater amoeboids, example the amoeba proteus, have contractile vacuoles. So we know that what is the function of contractile vacuole? It helps in the, maintaining the osmolarity. Right? So basically, it collects the excess water from the cytoplasm, collects before the vacuole. So before it feels that the vacuole is contracting, it collects the water from the cytoplasm, releasing the water through a temporary opening in the plasma. So when so it helps in maintaining the osmolarity, and therefore it helps the cell prevent it from getting shrink or from the burst or to burst up. So these are the functions of the different appendages or the different structures that are present in the structure of amoeba. So the figure here shows the amoeba proteus. This amoeboid is common in freshwater ponds. Bacteria and other microorganisms are digested in the food vacuoles, and contractile vacuole gets rid of the excess water. Just like eukaryotic cells, it has other membrane-bound organelles like mitochondria, and it has nucleus that contains a nucleolus as well, and surrounded by a plasma membrane. The cell is surrounded by a plasma membrane. The important thing to remember is the example and the structure, that which is pseudopodia. So this pseudopods helps in the ingestion of the food by taking the food inside the cell by the process which is known as the phagocytosis. So basically, they are heterotrophic in nature. They phagocytize their prey. That may be algae, bacteria, or other protists. The other ones are the flagellated protozoans. So amoeboid protozoans are not flagellated. Instead of flagella, they possess pseudopodia. While the other class is the flagellated protozoans that consist of flagellum. Right? So the membrane of this group are either free living or parasitic. They may exist freely in the nature, or they are dependent on the host for their food, survival, or nutrition, and therefore they are parasitic in nature. They have flagella, as the class suggests. They are flagellated protozoans. Example include the most important thing in flagellated protozoan to remember is the examples. These examples are very important for you to remember, as these organisms causes diseases which are very deadly. So the examples include Trypanosoma brucei, that is transmitted by the bite of tsetse fly, which is the cause of African sleeping sickness. So this is very important to remember. Example of Trypanosoma brucei, which is a protozoan, which is that belongs to the kingdom Protista, and it is transmitted by the bite of. This also you need to remember that how it gets transmitted. So it is transmitted by the bite of tsetse fly, and it is the organism that causes the African sleeping sickness. Similarly, we have another trypanosoma, which is Trypanosoma cruzi. So we have Trypanosoma brucei that is caused by the bite of tsetse fly that causes African sleeping sickness. We have Trypanosoma cruzi that causes Chagas disease in humans of Central and South America. So C you may remember that Trypanosoma cruzi causing Chagas disease. Trypanosoma brucei causes African sleeping sickness. So this is an example, or this is a micrograph of the A figure represents a micrograph of the Trypanosoma brucei, which is the causal agent of African sleeping sickness among the red blood cells. So you can see how they are attached to the red blood cells, or they are present near the red blood cells. The other figure here, the B figure, represents the general structure of the Trypanosoma brucei that contains a flagellum. The other structures will be cell like the other eukaryotic cells, and they have an undulating membrane. That is, they have a folded membrane that also facilitates in the movement and along with the flagellum. So along with the flagellum, undulating membrane, this cover is present around the trypanosoma. This is the membrane which surrounds the trypanosoma, which is the undulating membrane that is present. So the most important thing to remember is that in flagellated protozoans, flagella are present. They may be free living or they may be parasitic. Examples you need to remember that examples include Trypanosoma cruzi that causes that are transmitted by the bite of CC fly that causes the African sleeping sickness, or Trypanosoma cruzi that causes Chagas disease in humans of Central and South America, killing many people every year. The next is the ciliated protozoans. So in ciliated protozoans, they have cilia for the movement. So they are unicellular protists and they use the cilia for the movement. Structurally, they are most complex and specialized of all the protozoa. They form a diverse group of protozoans ranging where their size ranges from 10 to 3000 micron and the number approximately 8000 species. Most ciliates are free living. However, several are parasitic, that is, they are dependent. Sessile means they are attached to some other organism, and or they may be colonial. Members of the genus Paramecium are classical examples of the ciliates. Most ciliates ingest their food. For example, when a Paramecium feeds, food particles are swept down to a gullet, below which the food vacuoles form. Following digestion, the cytoplasm absorbs the soluble nutrients and the non-digestible part is eliminated at the anal pore. So this, we can see the diagram here. We have an anal pore to which, so we have an anal pore and we have, so this is a diagram of Paramecium where we have contractile vacuole, we have structures like contractile vacuole, we have thousands of cilia that cover the surface of Paramecium, we have oral groove through which the food enters, we have a cell mouth, which is the, then through the gullet, the food enters into the food vacuole. Food vacuoles fuses with the lysosome. So this is the food that enters, right? This food vacuoles fuses with the lysozyme and in the presence of, since the lysozyme contains enzymes, so this helps in the digestion of the food. As the food is digested, the vacuoles follow a looping path through the cell. So they form a looping path through the cell. Waste are released when the vacuoles fused with the specialized region of the plasma membrane that functions as an anal food. So this is how the food enters. Through the cell mouth, through the oral groove, where the food vacuole fuses with the lysozyme, the food digestion takes place here. So the food digestion takes place here in the presence of the lysozyme, enzymes that are present in the lysozyme, and then it follows a looping path, and the food vacuole basically, ultimately the vacuole that contains the waste is then fused with the plasma membrane, releasing the waste outside the cell. A very important thing is that it has two nucleus. One is a micronucleus and the larger one is a macronucleus. So the macronucleus controls the normal metabolism while the micronuclei is controlled with the reproduction. So this helps in the reproduction while this is concerned with the reproduction while the macronucleus is concerned with the metabolism. Now we have contractile vacuole also. The contractile vacuole, as we know that the function of contractile vacuole is to maintain the osmolarity by the process of osmosis from its hypotonic environment. It allows constantly taken water. So it is really important. So Paramecium when present in hypotonic environment, as I mentioned, when its organism is present in hypotonic environment, water will keep on entering into the organism, due to which there will be a time when the organism will burst. Therefore, it is really important to maintain the osmolarity, which is maintained with the help of the contractile vacuole. So, bladder-like contractile vacuoles accumulate excess water from radial canal and periodically expel it through the plasma membrane. This is how it maintains the osmolarity. So, this is the different structures that are present in the Paramecium and their function. So, the important thing is that dinoflagellates, we have two flagella. In ciliated protozoans, we have two nuclei, larger nuclei and a smaller nuclei, macronucleus, micronuclei. Macronucleus help in the metabolism and micronuclei help with the reproduction. The last the type of protozoan that is described is sporozoans. Sporozoans are nearly 3,900 species. They are non-motile, they are parasitic, they are spore-forming protozoans. They include the organisms that have infectious spore-like stages in their stage. For example, very important example is Plasmodium. Examples are really important in classes of protozoans because they are the ones that are causing the diseases to humans. So the very important example is Plasmodium. Plasmodium that causes the malaria. So it is the malarial parasite that causes the malaria, a disease that has a staggering effect on the human population. So we know that Plasmodium at some stage of their life produces spores. These sporozoids or the spore-like stage is infectious and can cause malaria, leading to the sometimes being fatal to the humans. So this was about the protozoans. We can just quickly go through them once again. So protozoans are amoeboid, for example, amoeboproteus, flagellated protozoans like Trypanosoma cruzi and Trypanosoma cruzi, ciliated protozoans, for example, Paramecium, and sporozoans like, for example, the Plasmodium falciparum, Plasmodium vivax, that are the malarial parasites or the disease-causing Plasmodium that causes the malaria. The next is slime molds in Kingdom Protista. So in Kingdom Protista, we first learn about Chrysophyta, then we learn about Pyrophyta, then we learn about euglenoids, then we learn about protozoans, and the last we will learn about is the slime molds. So slime molds are saprophytic protists. So they are not autotrophic, but they are saprophytic. So slime molds are saprophytic protists. They are found in forest and woodlands, and they contribute to ecological balance when they phagocytize and therefore help dispose of the dead plants and animals. They also feed on bacteria, keeping their population under the control, and they were once classified as fungi, but unlike fungi, they lack cell walls. So they were once classified as fungi, but they lack the cell walls. While the fungi, we know, have a cell wall which is made up of chitin, which is also known as fungal cellulose. And they have flagellated cells at some time during their life cycle, unlike fungi. The vegetative stage of the slime mold is mobile and amoeboid. So we have two stages in the life cycle of slime mold. One is a vegetative stage, one is the reproductive stage. So the vegetative state of the slime mold is mobile, that is, it has the ability to move, that is, it is mobile, and amoeboid. So we can see the structure, which is amoeboid-like structure, which does not have a well-defined shape. And slime molds produce spores by the process of meiosis. So basically, it's kind of sexual reproduction. The spores germinate to form the gametes, which then these gametes then undergo fusion, forming a zygote and resulting into a newer organism. So we have two types of slime molds: plasmodial slime molds and cellular slime molds. So plasmodial slime molds exist as Plasmodium, which is a diploid that is to an multinucleated cytoplasmic mass enveloped by a slime sheath that creeps along, phagocytizing the decaying plant material in the forest or agriculture field. Approximately 700 species of the Plasmodium slime molds have been described, and many of these species are brightly colored. So this is the reproductive cycle of the slime mold. At times unfavorable to growth. Like, for example, suppose this is a mature Plasmodium, right? Plasmodial. So do not confuse this Plasmodium with this Plasmodium. This is a Plasmodium which is a sporozoan. This is a Plasmodial slime mold which is different. So this is a mature Plasmodium. The Plasmodium here is the stage. So this mature Plasmodium, suppose during the time of unfavorable growth, like for example drought, it grows into the sporangia. So it develops many sporangia and sporangia formation begins. So a sporangia is a reproductive structure that produces the spores. So sporangium contains the spores. So this is a young sporangium, and an aggregate of sporangia is called a fruiting body. So fruiting bodies are, we can say that fruiting bodies are formed. The spores are then produced by a plasmodial slime mold sporangia. So basically these young sporangium undergo meiosis. This is a mature sporangium that releases the spores. This is a germinating spore. So under the when there are favorable conditions, the spores that are being produced by the plasmodial slime mold sporangium, when they get enough water, enough moisture to survive, which are sufficient for their germination, they germinate. So this is a germinating spore that produces the cells. The plasmodium slime molds spores release a haploid flagellated cells. So the spores can release a haploid flagellated cell, or they may release an amoeboid cell. Amoeboid cell-like structure do not have flagella, while the flagellated cells do have flagella. In the later stages, these two cells undergo fusion, and upon fertilization, they form a zygote, which upon development results in the formation of young plasmodium, which upon further development results in the mature plasmodium. And the cycle continues. So till young sporangium, the stages of zygote, young plasmodium, mature plasmodium, sporangia, and young sporangium are diploid in nature. That is, they are 2n. So this stage is diploidic in nature, while they undergo meiosis. So young sporangium undergoes meiosis, and from 2n to n, it converts to n. That is, these stages are haploid in nature. The next is the cellular slime molds. So unlike plasmodial slime molds, cellular slime molds are so called because they exist as individual amoeboid cells. So they are common in soil where they feed on bacteria and yeast. Nearly 70 species of cellular slime molds have been described. During unfavorable conditions, the cells release a chemical that causes them to aggregate into the pseudoplasmodium. It's not a true plasmodium, but a pseudoplasmodium. The pseudoplasmodium stage is temporary and eventually produces a fruiting body in which the sporangia produces the spores, as we see in this case. So in this, the structure is pseudoplasmodium, and then when the favorable conditions return, the spores germinate, releasing haploid amoeboid cells, and the asexual cycle begins again. So in the cellular slime molds, we do not have any it is mainly through the asexual reproduction. So it happens when the favorable conditions return, the spores germinate, it releases the haploid amoeboid cells, and the asexual cycles begin. So this was about the cellular slime molds. So this is the ending of the kingdom protista. We will learn about the kingdom fungi in the next session. So thank you for attending it.